So very good morning, my dear students. This is the new semester, the second year, second semester. In the second year, second semester, so you are going to study a course, Basic Microbiology and Phytopathology from the Department of Agricultural Biology. So this is a 3-credit course. That's what it is indicated in yellow, 3-credit course. And the second course, it is indicated in red. So the second year, second semester, so it's indicated in the dark blue and the purple color. So we can move this course, then introduction of the course, then we will continue with the importance of the microbes to the agriculture. So in the course is encompassed with the coupled with the 2 branches. So one branch is we call it as in basic microbiology. The other branch is we call it as phytopathology. So actually the phytopathology is a branch of the microbiology, but for the easiness of the course, we are dividing this one as basic microbiology and phytopathology. For the agricultural point of view, so your main task would be phytopathology from the Department of Agricultural Biology, but the basic microbiology covers the characters. So the microorganism, what are important to agriculture? It's a broad point of view. So you do some microorganism you may study in the animal science, some microorganism you may study the food sciences. So the agricultural point of view, so basic microbiology will cover the importance of microorganisms to the agriculture and the microbes and their classifications. How you are going to classify the microorganisms, so based on the different morphological and physiological differences, and the growth and reproduction of these each and every microorganisms, how they grow, what are the substrates and the conditions needed for the growth and development, and in which host they are selecting to reproduce, or which host or medium. Some organisms you can culture in the artificial medium. So in which medium and what are the conditions needed for the reproduction of these specific microbes. And symbiotic nitrogen fixation. So you all know the microorganisms involved for the fixation of the nitrogen. So symbiotic nitrogen fixation is very, very important to the agriculture. So therefore, how these symbiotic nitrogen fixation is happening in the plants, what are the organisms involved in the nitrogen fixation. And other chapter you will study as an effective microorganism and its importance to the agriculture. The effective microorganism, you all know, these are the group of microorganisms, so especially selected by a scientist, the Japanese scientist, and he made a solutions called effective microorganism EM solutions. This EM solution consisting certain different groups of microorganisms that are very beneficial to the agriculture in various aspects. For the decomposition, to increase the soil beneficial microflora, and also to recycle the nutrition, to control the plant pathogens in different aspects. The applications of this effective microorganism is being followed in the agriculture. And other important aspects in the microbiology is the mushroom production. You all know the microorganism, mushroom is a microorganisms. So the mushroom production and its beneficial, the profit and the different types of mushrooms, how you can cultivate these mushrooms in the cottage and commercial level, and the microbial industrial technologies. So this is the small section we will cover, but in each and every industrial application, you will study under the specific course design in different departments. For example, for the yogurt preparation, what are the microbes involved for the yogurt preparation, you will study in the animal science. Right? What are the microorganisms you can use to produce insulin? Right? For example, certain bacteria, this means we call it as a transgenic bacteria nowadays used by the pharmaceutical industries to produce these certain antibiotics and insulin and other things, right? That section you will study sometimes in under the food microbiology, drugs, under the drugs conditions, right? Drug and its administration. So these are the section will be covered under the microbiology. And in the plant pathology sections, you all know the plant pathology is a science of studying the pathogens that are causing the disease on plants. That is we call it as a plant pathology. So under the plant pathology, you will study what are the different group of microorganisms causing the disease on plants, especially crops. So plants is, and you all know this is a broad concept. The crops is we call it as a domesticated, plants we call it as a crops for the human needs, human and animal, right? Animal husbandry and the human beings. So if we domesticated certain plants, that's what we call it as a crops. So introduction to the plant disease mean how these organisms that are causing the disease, and when they are causing the disease, certain process will happen for the successful disease development. That is called as a stages of the disease development, right? From the coming and the contact and the initiation of these different the host pathogen recognition. Once the host pathogen recognizes successful host, the pathogen will penetrate. So then it will invade it. Then it will absorb the nutrition from the host. Then it will growth and multiply. And it is that's why in the way this life cycle of the bacteria or the fungi or the nematode or protozoa is complete. And finally, at the plant will react. This one you will observe these reactions, it will be exposed as a disease, right? So symptoms, especially the reaction we call it as a symptom. So that's what the plant will become, will not be a normal condition. If the plant is not normal, we call it as a plant under disease, right? Plant under disease. That is we call it as a stages of disease development. Certain stages, different stages. You will study detailing the stages of disease development. And effects of pathogens on plant physiological functions. You all know, so for example, if you are going there, if you are fever, you are having fever, right? If you are going there in the doctor, he will ask to do this urine test, blood test, this test and that test and different tests. Why? So because their pathogens are targeting on the physiological function of the host. Here you will study what are the plant physiological function, what are the, you all know, you under the first year, second semester, you would have studied a lot of plant physiological function, photosynthesis, water translocation, food translocation, transpiration, protein synthesis, and different plant physiological functions are there. Then how this plant pathogen interfere with the plant physiological function. If it is interfering the plant physiological function, so plant will get disturbed. If the plant get disturbing, then the plant will not normal. If the plant is not normal, we call it as plant under disease condition. So that how these plant pathogens affect the plant physiological function. You will study. So different plant physiology and how these different plant physiological functions are interfered or intervened by these plant pathogens. Plant pathogens are a group of microorganisms, right? Group of microorganisms. And the gene for gene concept. So gene for gene concept is related with the host genetics. Host genetics and the pathogen genetics mean, for example, if a pathogen is, if a microorganism, we call it a pathogen, right? Plant pathogen. Or phytopathology. So pathogen is selecting the host. So because of the certain preferences of these microorganisms or pathogen, this pathogen, how they are selecting the host means certain genes involved in the pathogen. So that is recognizing the signal producing by the host plant. The signal, how this host plant producing signal, maybe the proteins, maybe the chemical. Because of these genes present in the plant encoding that proteins or chemicals. If the pathogen is sensing that chemicals, And if this sense chemical is overwhelmed by the pathogens, then that host will be selected by this pathogen as a for the food to grow the nutrition and growth and reproduction. That is depend on the gene for gene concept. So whatever the gene present in the pathogen and whatever the gene present in the host, so interaction of these certain genes helping for the successful development of the disease. Or if the gene present in the plant is very dominant, we call it as a resistance. Meaning it will not allow the plants to if the plants will not if the plant is more resistant, it will not allow the pathogen to get in. That's what some of you are very resistant. Some of you are very susceptible. If you are going under the rain, so in next day you are sneezing, you will feel headache, fever because you are susceptible. The pathogen overwhelmed you and it's made you as sick. Right? That is, you will study for host pathogen genetics under the gene for gene concept. And then you will study the plant defense mechanism. If the host pathogen genetics is explaining how the plant, host is resistant. And also, if the host is resistant, it will defend against the pathogen. And if you are going there in the road, some robbery is coming. If you are a very vibrant person, a very courageous person, you will defend against the robber. This we call it as a defending. Right? So if the defense means the host is a very resistant. The host side we will talk always resistance. The pathogen side we will talk always virulence. Right? It's virulent. Pathogen side is a virulent. Host side we will talking about resistance. Right? And also environmental factors favor the disease development. So environmental factor, you all know if the pathogen is there, the host is there. In the presence of the pathogen and the host in every time, right? The pathogen of the host and present in the, right? Pathogen in the host. Pathogen of the host is interfering, right? So in always, maybe the susceptible host or maybe the virulent pathogen, they are interfering, you will not expect the disease all the times. The third factor will influence, we call it as an environmental factors, right? So if the presence of the environmental factor in certain condition, in the certain time, only you can expect the disease. For example, the human disease, you will. In the humid condition or the rainy season, you will expect a cough, sneezing, fever. At that time, you will not expect any type of chickenpox or the smallpox or these viral diseases. When the temperature is increasing, you will not expect any sneezing or fever. You will expect this this smallpox or the viral disease, all the viral diseases. Why? So because of these environmental factors. For the fungal disease or bacterial diseases, it needs moisture. That's what in the rainy season you are getting disease of the bacterial and fungal disease. So viral diseases normally you can expect in the dry season, dry cough, sneezing. That is mainly because of the virus, not the bacteria or the fungus. Right? It means so certain environmental factors are important for certain group of pathogen and also certain group of hosts. So that's the things we call it as an environmental factors favor the disease development. That is, you will study under the disease triangle and disease tetrahedron. So this is in theory explained by the scientist, right? How these environmental factors interfering when the host pathogen interacting. The host pathogen is present, so the third factor will interfere to make a successful disease development. So when the disease is successful or the pathogen is the host is get infected with a certain pathogen, right? this infection will be, the host will defend it, or the pathogen will influence the host. The host will show certain expressions. These expressions we call it as a symptoms. For example, the COVID-19 attack, the first symptom is you will get fever. It means sneezing, dry cough, pain on the throat. This is we call it as a symptoms because of your body is reacting against that pathogen. That's the way the disease symptoms are. So if the host is reacting against the pathogen, it will show certain symptoms. That's what you will study certain symptoms. For example, little leaves in chili, rotting of potato, Phytophthora. Right? Banana, you mean anthracnose. In the rainy season, if you're going there in the any shops, you will get the banana with blackish symptoms. Banana fruits with a black symptoms. It's called anthracnose. This is because of this disease. In the disease symptom, you can expect certain pathogen. That is we call it as a sign. For example, if you are going there in a shop, fruit shops. So from today, if you are going to a fruit shop, you can see, right? Papaya, especially papaya. The rainy season, if you are going there in the papaya, you can find the papaya fruit certain symptoms. Above the symptom, you can find white color mycelium or green color mycelium or blackish color mycelium with a spores. Or if you are at home, you are keeping the onion or potato for a long time, you can find the rotting of potato with a white color mycelium. If this pathogen part is present in that symptom, we call it as a sign. That's what it's differentiating sign and the symptom. Sign is a physically presence of the part of the pathogen or the pathogen spores or any mycelia, anything, we call it as a sign in the symptom. Symptom is a visual expression of the host, a visual expression of the host due to the pathogen attack. That's what we call as symptom. If you are collecting the symptoms definitely all the time you will not expect a pathogen. But if you are keeping under certain environmental condition, you can observe the signs. That is, you call it as a diagnosis. How you are going to diagnose a disease from the symptoms and sign, and also using this microscopic examination and advanced plant disease diagnosis techniques, biochemical test, physiological test. So nowadays the molecular techniques has boomed. So through the molecular markers, PCR-based diagnosis, right? PCR-based. And in the quarantine, right? In the quarantine, so not in Sri Lanka, in the well-developed countries, now they are using that fluorescent image detection systems. So if you are scanning that vegetable or potato or anything under this system, it will indicate that whether the pathogen is present or not. It's an advanced image processing systems. And also certain infrareds they are using. It's an advanced techniques. Nowadays it's emerging, right? Emerging advanced techniques to diagnose the disease quickly, right? Loop amplified thermal reaction. So there are different techniques, and you will study under this. And finally, you will study the integrated disease management. How you are going to manage the disease using the whatever the techniques available or practiced by the farmers or invented by the scientist. We call it as an integrated disease management. So nowadays, so the integrated disease management, mainly they are focusing on integrated plant soil health, right? Integrated improvement of plant soil health, right? So that's the way they are now focusing, because the 2020 is a year of plant health. UN announced that 2020 is a year of plant health. Year of plant health means how you are maintaining or improving the health of the plant It's mean planting material, seed or any plants to get optimum production, optimum yield. If you are sick, your productivity you cannot expect it your maximum. That's why the plant, if the plant is healthy only, you can expect the maximum output from the plant. That is so very vital, very important for food security. Otherwise, you will not get enough food. No? So now the population is increasing. So now it is 8 billion, right? So in 2030 they are focusing it will reach 10 billion, 100 billion. The 17 billion in the 2100. And how you are going to feed the populations? So you have to care on this plant health. Otherwise, we will get lost, right? Because of these environmental changes. So these are the sections. So will be covered under this basic microbiology and phytopathology, right? So the microbiology, right? We are going to now discuss detailedly about the sections under the basic microbiology. So the microbiology is a science, right? Microbiology is a science. It's a dealing or study of the microorganisms that are, we call it as an organism that are very small and cannot see through your naked eye. We call it as a microorganisms. So the study of these very small microorganisms, we call it as a microbiology, right? So the microorganisms, commonly they call it in different names, right? Some people some literature it says they call it as germs. So some in literature somebody may can write as a bugs, right? It's include bacteria, viruses, fungi, algae, protozoa, helminths, pyrons, MLOs. So they are different categories of organisms. So that organism you could not able to see through your naked eye. But certain reproductive part you may see under this. For example, if the fungus, under the fungus is mushroom, you can see the mushroom fruiting body and some of the mycelia. But actually you cannot see the spores of these microorganisms, or the fungi. So that's what we call it as a microbiome. The fungi also under the microorganism because you could not able to see under the your naked eye, right? So these branches of microbiology is maybe so there are different branches. You can say I told you in the first slide also, right? So microbiology is a very wide section. Under these sections, you can study, you can find it. So bacteriology, mycology, virology, parasitology, immunology, pathology, right? Pathology. So different branches. If you are going there in the agriculture university, for example, if you are going to Tamil Nadu Agriculture University. So this is a Tamil Nadu Agricultural University. There is no other university. It is not. There is no any agricultural university in Sri Lanka. Right? If you are going there in Pakistan, you can find several agricultural universities. Right? In the agricultural universities, under the Department of Agricultural Biology, or sometimes some departments are there, Department of Microbiology, or Faculty of Microbiology. Under the Faculty of Microbiology, you can find a Department of Bacteriology, Department of Mycology, Department of Virology, Department of Parasitology, Department of Immunology. For example, if you are going to the medical faculty in Jaffna University also, you can find the Department of Parasitology, Department of Helminthology. So helminthology also is under the microbiology. Helminthology means dealing with studying of nematodes. Mycology is a fungi, virology is virus, Parasitology is some of the protozoans. Immunology means certain pyrons and other things, they are causing the allergens and diseases. The humoral cellular immune responses. Nowadays, people are getting VC. So nowadays, people are getting some kind of sneezing, some of kind of unknown diseases. So because of allergic reactions, right? Formation of tumors. This is governed by those are the sections covered under the immunology section. So, but here you will not study the bacteriology, or virology, or mycology, or parasitology. You will study these microorganisms, different microorganisms under the old coming under the old logies, bacteriology or mycology. You will study about bacteria, fungus, and virus. You will not study about parasitology or immunology or helminthology. But some sections, some part will be covered by from the helminthology, right? But for agricultural point of view, we need a basic concepts, right? So you will study the bacteria and this classification of the bacteria and shapes and the function. Viruses in the classification of the viruses and the shapes and the function, importance in agriculture. And fungus, its shapes and function and importance in agriculture. Those things you will study under this basic microbiology. The specialization in microbiology, I told you, so there are different agricultural and veterinary microbiology. So if you are going there, if you are planning to do postgraduates, you're having the basic agricultural degree, you can specialize on those sections and you can get a postgraduate degrees. For example, food microbiology, master in food microbiology, PhD in food microbiology. So epidemiology and public health microbiology. Then you can work as a public health inspector from the basic agriculture. It doesn't matter whether you need a basic degree. Even doctor you can study in the medical microbiology. There are also scopes, but you need a basic degree. If you are feeling that you could not be able to enter the medical faculty, but these sections, so if you are having the basic degree, you can specialize, but you can't be a doctor, medical doctor, but you can work under the with the medical professionals, right? Work with a medical professional as an for health microbiologist. And food and drug administration, you can work food microbiologist. In veterinary microbiologist, environmental microbiologist, right? And so this is why we are studying this microbiology is because these microorganisms, so they are very, very important, not only the agriculture, this whole world depends on the microorganisms. So microorganism can cause the disease, or this means we will study at the harmful effect of microorganisms. So there are other, instead of this detrimental effect, there are a lot of beneficial effects we are getting from this microorganism. The first benefits we are getting from the agricultural point of view, the food production point of view, means fixation of atmospheric nitrogen. The fixation of the atmospheric nitrogen is, so every day you are producing millions of tons of nitrogen as an ammonia. This is consuming a lot of energy as the fuel energy, the human and the investments. But micro and also certain, you have to develop a certain condition. So 400 atmospheric pressure and the high temperature only you can get the ammonia. But nitrogen is abundant in the atmosphere. If the microorganism can able to fix that nitrogen into ammonia freely, then why we need to go for an industrial nitrogen production? For the eco-friendly way, the atmospheric nitrogen can fixed by these microorganisms. If you are focusing on the researchers to modify these microorganisms in a way to increase its efficiency of fixing nitrogen, then you need not to produce industrial ammonia, industrial nitrogen fertilizers. government need not to give the subsidies for the farmers. How much billions of dollars we are spending for giving a subsidy to the farmers in the ammonia subsidy, fertilizer subsidy. Why? Because it is produced under foreign countries. For the production of the 1 kilogram of ammonia, it needs more than 6 liters of oil. How much of carbon dioxide this oil, burning of oils produced? Environmental problem. But this microorganism is there in everywhere if you are inoculating. So it is there in the nature. The problem is if you are applying the pesticides, so it will kill these microorganisms or reduce the efficiency of these nitrogen fixing microorganisms. That is the problem. That's what we are emphasizing, to go back towards organic agriculture. Right? So fixation of this atmospheric nitrogen is one beneficial effect. You will study under the nitrogen fixation by the microorganism, and decomposition of the organic waste and residues. So in every day, in every household, how much of waste you are generating. If you are generating continuously, if it is not decomposed properly, this world is full of waste. The microorganisms, there are a group of microorganisms. That's what I told you, one of them, one group of microorganisms made by scientists, Japanese scientists, we call effective microorganism. So these effective microorganisms quickly decompose this whatever the materials, decomposable material. But the problem is our people are mixing this decomposable material with non-decomposable polythene and other things. If the decomposing materials are there, quickly you can decompose it. Everywhere the solid waste is very big problem. Liquid waste is problem. But liquid waste, no problem. The people, what they are doing, just treating with the chlorine or the ozone, and they are leaving in the ocean. But solid waste, how? You have to recycle it. Again, you have to use it for agriculture purposes. Right? So the organism, microorganisms, so thermophilic microorganism and mesophilic microorganism. So there are a group of microorganisms. That's what you will study. Sometimes it will be covered under the soil and soil fertility, the compost making. And also we are also doing the vermicompost production and the mushroom worm indicated compost production, right? 4 to 5 per month, the compost productions in our Department of Agriculture Biology, right? So decomposition of the organic waste. It's the benefits of the microorganism. And suppression of the soil-borne pathogens. Certain group of organisms, for example, Pseudomonas fluorescens, Trichoderma. Pseudomonas fluorescens is a bacteria. Trichoderma, different species of Trichoderma harsianum and viridae and other things. So Trichoderma pallidum. So these species are suppressing the soil-borne pathogens because they are producing certain antibiotics or certain chemicals. That chemicals will not allow this bacteria, other plant pathogen to touch with the host. It's a kill. Or certain microorganisms produce certain antibiotics, it will kill. That antibiotics we are using now, especially the fungal producing antibiotics we are using and the pharmaceutical industries, right? This amoxicillin, augmentin, cephalexin, cefixin, the different antibiotics. Yeah? So because these are produced by these microorganisms. And recycling increase availability to the plant nutrition. So they are, while they are decomposing these material and also to the recycling this microorganism and conversion of these waste into usable nutrients, ammonia. So nitrogen into ammonification, the ammonifications, right? The nitrogen cycle and carbon cycle and phosphorate cycles. So VAM, vascular microorganisms. These organisms, so what they are doing, so making that waste, recycle that waste, increase the availability into to the plants, nutrition availability, especially in the forest. Are you adding any fertilizer to the forest? Even how the plants are flourishing nicely, growing nicely. So because of this recycling and increasing availability of the plant nutrition from the waste produced by the same forest trees. Right? And degradation of toxic. Certain microorganisms, pathogens are producing toxins. This if the group of beneficial microorganisms are there in the soil, so easily they will detoxify these toxins. And also certain heavy metals, pesticides residues, a certain microorganism can able to degrade it, detoxify that. And production of antibiotics and biotech compounds, I told you, right? Secondary metabolites we are using as a pesticides and herbicides. And certain antibiotics we are using as for the human drug, or we are using against plant pathogens. And simple organic molecule, production of simple organic molecules for plant uptake. For example, VAM, vascular mycorrhiza. So in the Epavala rock phosphate, in the rock phosphate is immobile form. But the VAM fungi is making it available from PO4 3- form. It's a rock mineral phosphate, the PO4 3-. So do we need to import phosphate anywhere from the, because it's the Epavala is the Andhra district is rich of this Epavala rock. So government is now thinking to making the phosphorus fertilizer by using these microorganisms into phosphate fertilizers. So you cannot add directly to the mineral phosphate to the plant, so you have to convert it. That's what you need microorganisms help. And solubilization of insoluble nutrients. Certain nutrients will not soluble. For example, the certain ferrous, the iron, right? Certain sulfur components are insoluble. So it need to convert in a different form to get the solubilization. That's what certain microorganisms are involved. And complexation of the heavy metal into limit uptake of plants. So if the heavy we call it as a metal chelation. You will study in the soil, right? So metal chelation means certain heavy metals are there in the soil. So it is, if it is absorbed by the plant, it will directly reach to the human being. But what the microorganism making these metals into through a metal chelation into unabsorbable form by the plant. Plant cannot absorb. So both ways we are doing, we are solubilizing and taking into the other way around. And the production of the polysaccharides to improve the soil aggregation. Right? So the certain microorganism producing certain their enzymes as a secondary metabolites. See, secondary metabolites, if it is there in the soil, it can aggregate the soil particles and improve the soil structure and texture. Not the structure, texture. And food for the human and livestock. So every day, certain countries, we are using the microorganism as a food. For example, marmite. So marmite is a product of the yeast, pure product. We are eating microorganisms. And also for the human food production. So yogurt production in the bakery industries, for the fermentations of vinegar and wine productions, microorganisms are involved. Otherwise, you cannot convert it. And certain microorganisms you are eating directly, as in, for example, these biofertilizers. These biofertilizers, you can give it this to livestock, right? You can give this one to the livestock. For one, the few examples are there. Nowadays, you can use it as a spirulum. Right? You can use for that as a spirulina. So you can use it as a food for the human being, and also you can give it spirulina and the concentrated to spirulina concentrate to livestock, especially the poultry pellets. There's poultries and the livestock. And also certain you can use as a biofertilizer. Certain microorganisms directly you can use as a alga. The blue-green algas, you can use it as a biofertilizer. Right? Azolla, especially the azolla. This azolla is also, you can give it to the livestock, food for the livestock in the dry conditions. So these are the beneficial effect of the microorganisms. So this is a picture showing how you are using these photosynthetic bacteria's help. This is a beneficial effect of microorganisms, especially these bacteria, photosynthetic bacteria present in the ocean, or where this man anthropogenic activities are not present, these bacteria are producing oxygen through their photosynthesis. In the way they are helping, and also nutrient, they are decomposing and helping for the nutrient cycle. And beneficiaries, this another one is the hopper, especially in Australia, right? Australia, the ore is there. It's pure copper, we call it as an ore. The gold is we call it as an gold particles are there in the pure. Copper particles are there in the pure. Sulfur particles are there in the pure, not pure. The old particles are not in pure. But we need this in the minerals. So if you want to get that copper and gold and uranium and from the mineral sulfide, those also are copper sulfide, gold sulfide, and uranium sulfides. We have to need certain microorganisms to make that one to minerals. For example, in acidothiophacillus and leptospirillum ferredoxins, they are converting the ferric and the copper and the sulfides complex component into mineral form. That's what these industrial, there are many industries, right? So the mining industries are depending on these organisms. And these food industries, synthesis of drugs, I told you, drugs, hormones, enzymes. Many enzymes, many people need because their organs will not produce sufficiently. But nowadays, these modified bacteria, right? Modified means genetically modified bacteria, means because of gene editing technologies, we have made that one. We inserted that gene producing the enzymes and hormones into the bacteria. So bacteria quickly replicate now in 48 hours. If they are multiplying, while multiplying, this gene will encoding this protein, they are encoding these enzymes and hormones. These enzymes and hormones are using the hi-fi techniques. They are filtrates collected. Nowadays we are using as an insulin. Certain people, so certain hormones will not produce, right? Especially thyroid hormones, it will not produce. So you need to give an extra administration, oral administration or intravenous injection. That's what all these enzymes and hormones are produced by the microorganisms. Certain proteins, especially certain proteins needed for the brain producing proteins are needed for certain disease people. How they are producing this protein, especially from these microorganisms. And also food industries depend on these microorganisms, yogurt industry, marmite industry, vinegar industry, wine industry, lot of industries. And waste recycling. Nowadays, scientists made a modified microorganism. Sometimes we call it as a superbug. Would have studies in advanced level. So the bioremediation, means cleaning that oil contaminated seawater, ocean water, the oil contaminated soils, and heavy metal contaminated soil. We call it as a bioremediation, right? Bioremediation means removal of the heavy metals and non-degradable material from the soil or water. We call it, using a microorganism, we call it as a bioremediation. For the bioremediation process, many microorganisms are involved. And biofuel production. Nowadays, biofuel, they are by using the many algae, people are targeting to produce biofuels. If they are producing the biofuels, means you can use instead of the other petrol and diesel and others. Certain microorganisms nowadays, people are trying their microorganism producing electric current in the air. So nowadays, the scientists are working on to how they can improve these electric producing microorganisms. So nowadays, you can use it in the watches and the earphones instead of battery. They are in the air, it is producing the current. If you are inserting the chips of these microorganisms, then it can produce the current for small devices. You need not to change the batteries and others. Other than this beneficial effect, you can expect a lot of harmful effects of microorganisms. One such example is a plant diseases. There are a lot of diseases. So plant, animals, and humans. But we will focus on these plant diseases caused by these microorganisms. This is a detrimental effect to the agriculture, especially some organism, food, Clostridium. The stimulation of the soil-borne pathogens, right? Certain microorganisms, what they will do is stimulate the soil-borne pathogens. Why? So because they will produce certain secondary metabolites. These secondary metabolites will attract soil-borne pathogens because soil-borne pathogens will sense it. This one has chemicals, so if the chemicals is there, the plant will be there. Definitely, it will stimulate the dormant microorganism to proliferate. Immobilization of the plant nutrients. So, there are some organisms they are mobilizing, some organisms are immobilized, converting to not absorbable. So that is a detrimental effect to the agriculture. Inhibition of the seed germination. Many organisms, so they are inhibition of the seed. That's what we call as a plant pathogens. Inhibit the seed germination. So that's what we have to treat that one with this the microorganism, or at the seedling planting time you will treat with the Captan and Theran certain soil treatment chemicals. Inhibition of the plant growth and development. So this is under the disease, especially in the viruses. Right? Production of the phytotoxic substances. So this microorganism produce toxins. These toxins are phytotoxic. Right? Phytotoxic material. So these are the harmful or the detrimental effect of the microorganism to the agriculture. Certain microorganisms, they are spoiling the food spoilage by producing these toxic substances quickly. In 1 or 2 days, you know the coconut, open it, right? So make it the 2 half, place it in somewhere else 1 or 2 days. You will find it's full of fungus. The bread, place it in an open place, really full of mucor and penicillium. Fruits, you cannot keep that for a long time. So because of these detrimental microorganisms, they're spoiling that. Therefore, so this detrimental effect of the microorganisms, so you should be careful on, and you should learn how to manage these detrimental organisms in different applications, right? So then, so infectious disease caused by these microorganisms. So the microorganisms, the first microorganism discovered under the discovery of these microorganisms, right? The first microorganism they observed, call it as an anthrax. So an anthrax is a virus. So sorry, the bacteria that is causing the diseases in animals and all the human. It can cause the diseases in human well as well. In 1876, the Robert Koch, right? Robert Koch, she proved that the anthrax is a disease caused by bacteria. From that, from the discovery of these microscopes, microbiology sector boomed and advanced, right? Boomed and advanced. And he is the first person who cultured this anthrax in the solid culture medium, right? From his germ theory, and he made a theory we call it as Koch postulates, right? These Koch postulates are everywhere we are using to diagnose this disease, not only the agriculture, everywhere, right? The Koch postulates indicates that, so he cultured the microorganism for the confirmation of the microorganism. He did this experiment. From this experiment, he developed a theory we call it as a Koch postulate. The Koch postulate theory explained that So if you are taking a sample, right, from the sample only you are going to confirm the disease. Now we call it diagnosis. Samples, organism should present. So therefore, that's what you are culturing in the laboratory and observing through the microscope or using or diagnosing by using the advanced diagnostic tools. Then, so you have to isolate the pure culture in artificial culture media, or if it is not culturable, you have to use the original host. Isolation of the pure culture. Again, you have to inoculate that organism into the healthy host. If the same symptoms develop in the healthy host, then again you have to isolate that organism from that same host, and again you should observe under the microscope, or you should diagnose these characteristic features. If it is characteristic features are similar to the first isolated culture, then it's a confirmation that this organism causes the disease. That is the explained theory by the Kush postulates. So that's what we are using this theory in everywhere in all plant pathology or microbiological studies for the disease confirmation, right? Disease confirmation. So in the modern uses of microbes, right? Modern uses of microbes are, so the microbes, whatever the microbes and whatever the microbes characters and everything you are studying, the microorganism are used in the modern sciences. For example, in biotechnology, nowadays a very booming sector of the science, we call it as a biotechnology. So in the biotechnology, using the microorganisms and these applications, so especially to modify the microorganisms and characterize these microorganisms using the advanced diagnostic tool, and transfer of this gene present in the microorganism to the other, or using these microorganisms as a media, right? Medium for the transfer, intermediary for the transfer. This is a science dealing with the biotechnology. In the biotechnology also you will study certain microbes. For example, Microbacterium tumefaciens. It's an organism everywhere using for the gene transfer. And also for the, by using this microorganism to produce a food and chemical. And DNA engineering technologies. Nowadays we are calling it as a COVID-19 is made by the DNA engineering technology, means that's the gene editing technology. For the gene editing technology or the molecular biology, you need microorganisms. So microorganisms protect. For example, certain isozymes to cut the DNA of any organism. So you are using certain isozymes. These isozymes are product of this microorganism. And curing therapy of the gene therapy. Certain people, they are facing troubles because of these genetic disorders. To cure the genetic disorders, nowadays, scientists with the gene sequences of all the microorganisms and the human gene sequences, means human biology, human genome is completely sequenced. All the chromosomes, all the markers present, all the nucleotide, single nucleotide polymorphism present, the genomes are now available. They are now comparing with microorganism. If there is a problem, they are trying that microorganism to clear this gene disorder or rearrange that order. But ethically, it's not allowed, but you need ethical clearance to do these researches. A genetically modified bacteria to protect the crop farms, pest and freezing. You see the BT brinjal, BT corn, BT maize, BT cotton. It means bacteria gene inserted into the plants. Right? Bacteria. Nowadays, what they are doing is to increase the vitamin A content. They are modifying the rices, golden rice. Now, increase the fat content of the certain rice cultivars. the omega-3, what they are doing there, they are getting the gene from the shark and trying to put in the certain group of plants to produce omega-3. So cold and only the fishes living in the cold conditions only they will produce omega, no? So that's for food fortification purposes. Nowadays, the microbes are needed to gene transfer in the modern sciences, biotechnology, or genetic engineering, or molecular biology. So that's all for the day. I am stopping here with. If you want to ask any questions, you can ask.